Welcome to HoTResDB. We hope that first-time users will find this tutorial useful for navigating our database. As described in the brief introduction on the right, HoTResDB features host transcriptional response data for VHF infection. All approved collaborators are able to log in to access their data. If you are a collaborator of the Conroe Lab, you are welcome to register for approval. However, you do not need to have a login to view publicly available data. All visitors are initially logged in as guests. The navigation bar provides access to the other web pages: Search, Data Stats, Help, and Contact Us. The Contact Us page provides visitors with a method of communicating with the HoTResDB team. All fields are required in order for the team to address any submitted issues. The Help page includes frequently asked questions. To view our answers, the user can click on the text of any of the questions. The Data Stats page includes information on all featured datasets. The table describes virus and strain, includes a link to the publication for more information, GEO or bioproject numbers, and links if available, the sequencing type for the data, the lab that performed the experiment, a collapsible summary, and additional information about the experiments. The search page is organized by several tabs. The tabs involved with querying the database are Search and Browse Database. Beginning with search results, all subsequent tabs are a higher level of processing of the one previous. In the Search tab, the user can select any of the search parameters listed for virus, strain, and data type, and the gene, and the database will be queried for all relevant datasets. The user can enter all of their genes of interest in the supported formats: Wikigene ID, UniProt ID, or Ensemble ID for either human or macaque. Identifiers must be separated by a whitespace character. If the user is not sure of the exact identifiers for their genes of interest, they are able to use the Browse Database search. In this search form, the user can choose the type of identifier they would like to search for. The options available are the following: part of the macaque or human gene symbol, part of the gene description, or a gene ontology term. The user can add multiple search terms and use a Boolean and/or operator to narrow down searches. Any of the search results can be added to the user's cart. If any are mistakenly added, the user can remove individual genes. To search for a new gene, the user must clear the previous search table using a button in the form. The user can remove additional search terms at any time. The contents of the cart can be deposited into the general search form. For this search, we'll be taking a look at the expression of these genes in the Ebola Mayinga and Lassa Josiah RNA-seq datasets. Upon submission, the Search Results tab is displayed. This table shows the human ensemble and Wikigene IDs for all gene hits. A link to the gene cards database is provided for each gene for users looking for more information. The Expression Data tab shows query results in numerical format. The first table shows expression pattern over days post-infection, and the second table shows log2 fold change over days post-infection. Either table is downloadable in CSV format. To visualize expression pattern over time, the user can view a line graph in the Counts Graph tab. Each data point provides a specific value via mouse over. To compare between specific genes within the results, the user can hide lines by clicking on their identifiers in the legend. For a better view of each graph, the user can open the graph in a new window. All graphs are downloadable in several formats as a function of the HoTResDB API. Additionally, all results can be sorted in two different ways. Genes per study, which shows each gene included in a particular study, or studies per gene, in which all studies are included for each gene. Alternatively, to look at everything relative to day 0, a user can use the fold change view. This view has all of the same options as the counts graph. The fold change data can also be viewed in heat map format. This view shows upregulated genes as red and downregulated genes as blue. The rows are clustered by gene. Columns are sorted in two different ways, by day post-infection or by dataset. These graphs are downloadable as well. We hope that this video has been a useful tool to help you navigate our database. If you have any questions, we are available via our contact page. Thank you.